Glycomics, well, you've probably heard of genomics and proteomics. Genomics is really the study of all the genes that are present and can be expressed in a particular cell. Proteomics is the same kind of thing, only it's looking at proteins. And glycomics is, again, related to that, but it has to do with all of looking at all the potential enzymes and proteins that are involved in glycan biosynthesis and breakdown, as well as proteins that recognize glycans. So it's basic in an information way. It's really trying to understand all the information that's encoded in the DNA to make glycans, to make complex carbohydrates, and to recognize them. So all of their structures and all of their functions and how they're made. So we have a research resource biomedical glycomics center here. It's part of one of the institutes of the National Institutes of Health, and we're really focused on developing technologies to be able to identify all these glycan structures and to look at all the genes of the enzymes that make glycans, so we can understand how they change when, let's say, a cell becomes a cancer cell. Now, the system that we're really focused on right now is understanding these glycan changes during the differentiation of stem cells, so embryonic stem cell glycomics. And that's really important because as we learn how to differentiate stem cells, we have to be able to always purify a specific population of stem cells. So when you take cells and you make them differentiate into another kind of cell, it's not always 100%. So you have to be able to remove the cells you don't want and to find the cells that you do want. So then you can grow those up and take them another step. So these cell surface markers that I've been talking about, the glycans, are very good candidates for molecules that we can exploit to separate out the cells we want from the cells that we don't want. So it's very interesting to the stem cell community, and we have stem cell biologists on our center and on our advisory board. It's very important for us to understand how these changes occur, because then we can develop antibodies, or we can develop other types of proteins to find those cells that we want. And that's one of the major hurdles in stem cell biology, that many or most of these cancers actually arise from stem cells. They are cells, in all of the organs in your body there are stem cells. I mean, you've heard of, they're easiest to understand in the cells in your bone marrow, for example. And leukemias occur because some of those stem cells in your blood, in the bone marrow, become cancerous and they start overproducing. But there's evidence now that may be true for, as I said, other types of cancers, that there is a stem cell population, very small amounts of cells in your organs whose job it is to divide and then turn into other types of cells. And that's sort of how your organs are maintained. If those stem cells become cancerous, not only do they make more cells, they really lose control. So they are really dividing very rapidly, and they're also differentiating. They're also changing into other types of cells as well. So the analogy that I like is of fire ants. I mean, everybody who lives in Georgia and does any kind of gardening or has a yard has to deal with fire ants. And there's a queen. So you can kill a mound of fire ants, but if the queen survives, you're going to, in a week, you're going to have another mound of fire ants. I mean, that's the way it works. So the queen is like the stem cell. So you can kill a cancer with radiation and chemotherapy and surgery or whatever you want, but if the queen remains, if the stem cell that you needed to get rid of that's become cancerous is not killed, then you're going to get another cancer. You're going to get a recurrence of the cancer, and just like you get a recurrence of a fire ant hill. So we need to really understand what these stem cells look like in tissues. And again, the cell surface markers are very important, because if we know what they look like and we know what their cell surfaces are like, and of course my ideas are that they're going to have a unique glycosignature on their surfaces, then again we can find antibodies, or we can find carbohydrate binding proteins to seek out and kill those cells. So that's the only way to kill a fire ant colony, is to kill the queen. So it's the same analogy. If you can direct the chemotherapy to the stem cell in the tissue, that's what you want to do. Because if you kill that stem cell that's cancerous, then you got a real shot at keeping the cancer from going back again. So that's tying together what we know about cancer, what we know a little bit about in terms of stem cell surfaces, and really what I hope we'll be working on in 5 years, which is on cancer stem cells and how they're unique and how we can kill them. There's some very effective cancer therapies now where chemotherapy is directed. It's molecular chemotherapy. It's directed via an antibody to a specific cell type, and it's very effective in killing the cancer. So if we can find these cancer stem cells and direct antibodies to them with chemotherapy, I mean, that's the analogy, is to be able to kill those. So that's kind of how it all fits together.